Our next speaker is Andreas Diacon from University of Stellenbosch. Welcome, Andreas.
Thank you, Carl, and hello, everybody. Thank you for inviting me. I feel very privileged. I'm also here for the first time. I already have a drug that works. So I'm going to tell you here how I got that far. So you all know this. I mean, we need new or repurposed new substances that have no resistance, low potential for interactions, have no QT prolongation, and can partner up with others to make that new universal regimen that we all need. So this is now an example of repurposing. And this is the beta-lactam class, the carbapenems. And we know that there is a high intrinsic resistance against this genetically encoded beta-lactamase in MTB. And that was tested in vitro a number of years ago. You see all the publications at the bottom. There have been anecdotal reports out there that as part of an XDR-TB regimen, such drugs were given and the regimens turned out well, but there was never a control group and there was certainly never a study where the drug was given as monotherapy. And as an advantage of this class, there's many marketed compounds, so you can walk in the pharmacy right out there and get 3 or 4 of these substances off the shelf with a prescription, and they're usually very safe and have been in use for many years. Preclinical evidence and much of this will also be put into perspective by the next speaker, David Barros from GSK, who has basically done the basic science work and is currently still doing more about it. So the MICs against the standard bacterium are there at the bottom, and we saw that there are 2 compounds. Both of them are registered, have relatively low MICs, and both of these MICs drop further if you combine them with clavulanic acid. Now, clavulanic acid isn't available on the market as a free substance, so we tested what happens if one combines it with amoxicillin as well, and the MIC seems to drop even further. So this, of course, opened the door for clinical development, trying these 2 carbapenems, meropenam as an oral compound and meropenam as an IV compound, together with the combination of amoxicillin and clavulanic acid. Now here one has to mention that there is no reliable mouse model for these substances, and also that the carbapenems are very unstable in solution, so even measuring MICs is difficult. So that's what we did. Proof of concept in TB patients. I mean, you can read the text, but basically what we do is we find people with these chest X-rays. Does it display? Okay, yeah. On the right side, I mean, on your left side at the top, you see that there's this kind of hole in there. That's what TB does to lungs. It causes these cavities. And then we hospitalize these patients and we make them cough overnight, and this is what they're coughing out, so something between 10 and 20 mLs of sputum we collect over 16 hours. And then we put that onto culture plates and we count the colony forming units, so we use time to culture positivity in liquid culture. And we do this with regular intervals over 14 days in hospital under strict observation. And then we measure the drop of the mycobacterial viable load in sputum. And the drug groups we used, 15 subjects each at the bottom there, meropenam 600 milligrams with augmentin 1 tablet and meropenam 2 grams IV with augmentin both 3 times a day. So this is a proof of concept design. So you want to be sure that if it fails, you never have to do this again. So you want to give as much as possible, and these are the doses that are the highest that you can give within the current registration standards for other indications. And there was a control arm with a current combination, standard combination. So these are the typical patients that we find. About 2/3 of them are males, which shows you in Africa it's mainly the females that do the work and don't have time to participate in such trials. BMIs, these are people that are in the lower age of having normal weights, and the bacterial loads are about what we see in such trials. And this is basically the result. This is now just the straight CFU counts on the left. You will see the CFU logs. Between 6 and 7 is what we usually find in untreated patients. And over time, you can clearly see that one substance worked very well, one worked, and the other one didn't. And unfortunately, the red line is the oral carbapenem compound, the black line is the IV carbapenem compound, and the green line is the control, and the control did just about as well as we would expect. I mean, that's what the control is there for. And these are the results now adjusted for baseline values in a statistical model, and it's very clear that the meropenam augmentin IV treatment on the left side, it's clearly more than 0, so this is a very clear positive proof of concept. Whereas the second block there, the oral compound didn't show anything, and the control did as expected. So we don't usually do statistical significance testing here, but on the models, it's clear that both the meropenam and the control were better than nothing, and the control was a little bit better than meropenam. So to put this into perspective with the pharmacokinetics, the curves were all very nice. Amoxicillin, clavulanic acid as expected. And at the top, you have to look at the scales on the left. You can see the meropenam is perhaps just about 2 logs less in the exposures. So, and that's probably the reason why it didn't work. We just didn't get enough into these people. And the meropenam had very high Cmaxes because of infusion. It's easy to achieve that. Something about adverse events. I mean, these are all approved compounds, so it's not really the focus here. And some bad things happened, like always. Somebody died from liver failure, but that was after the study when he entered a control treatment program at the clinic. One person had a deep vein thrombosis that was also under control. We believe that the rifampicin has something to do with this. And another patient we took off the trial because we weren't sure if he was just confused or had a bit of TB meningitis, but turned out to be nothing, but he still wasn't in the final analysis. Okay, the interesting thing here is the diarrhea at the bottom row, that it was equally frequent between meropenam and ferropenam probably shows that it's the augmentin, and it's not the carbapenem. If it was the carbapenem, you know, it should, because mero was given IV, it should be less or more, because the values were so higher. But that it's equal means it's probably from the augmentin. Okay, so in perspective with other drugs, this is now one of the things. You can only do historical comparisons. This is what I could find, the best ever recorded 14-day activity, and this is rifampicin 35 kilograms per kilogram that was tested by the Panacea Consortium. This is by far the highest ever recorded, followed by normal dose INH. Probably higher dose INH might be a bit better, it was never been done. And then we have this robust activity, 14-day activity of rifampicin, pyrazinamide, PA824, and bedaquiline, and meropenam augmentin fits right in there at the same numbers. All right, so this is a proof of concept for a new drug class. This is why I'm here. Again, thank you for inviting me. We know now that we can use this, but it has to be given IV 3 times a day. So this can be done perhaps in Europe in a hospital where people are isolated and such resources are available. I don't think this is for Africa, this is an option, unless it's a privately insured patient in a hospital. Now the question is if amoxicillin and clavulanic acid is really essential. I'm only asking this because having a bit of diarrhea is probably okay if you're in hospital, but if you're supposed to go back to work, which was most our TB program patients actually are, that might be a problem. So can we reduce this to once daily? There is ertapenem, that is an approved carbapenem out there. You can buy this. It's very safe, and it has high protein binding and a longer half-life, and it might just work or it might not. We've done some modeling, and some people think it will, some people think it won't, so the only way to find out is to taste it. And again, there it's going to be the question if you need augmentin. This is a compound that is approved for intramuscular injection as well. So it could actually be given in a program context. Streptomycin was given like this, kanamycin, amikacin is given like this every day. So this would actually then move it to an African setting. This compound could then be given there. Now, but the first prize is still an orally bioavailable agent. And there is pharopenem midoxamil is a compound that's very interesting. It's almost made it to marketing. There have been phase 3 trials done, and then somehow it was never marketed. And somehow the company that owned it is no longer around, and the patent has expired as well. So if any one of you has an investigator's brochure of that compound, I should have added my email address, but you can Google me, you'll find it. That would be very nice to have this, and try if we can't figure out somehow with an IV formulation. We would have to somehow do with the regulatory people, try if we can't find an oral dose of meropenam, and the medoxamil seems to produce higher exposures that will produce that carbapenem effect with an oral compound. This would really make it a TB drug that can be used in practice. Good, that's it. Perhaps a picture here, yeah, everybody that was involved in this, and the European Clinical Trials Partnership funded it, and some of these faces, one of them is somewhere here in the room. Yeah, but thank you everybody, you know who you are. Thanks for listening.
Thank you, Andreas. Any questions for clarification?
Hey, Andreas, thank you. So on the meropenam, that's very interesting. So I can say that with the meropenam medoxamil, which you can give up to doses of 40 mg per kg, the peak concentration actually gets very high. You get to be above your MIC for like a long time. It's worth the
Tawanda, it's a long question. Let me give you a microphone, because I think a lot of people can't hear.
Yes, I was just saying it's interesting. So in the hollow fiber system, the first of all, the ertapenem works very well, more than we expected for sterilizing effect. The meropenam actually works if you use the oral formulation, which they have given up to doses of 40 mg per kg before they disappeared, but they have their abstracts around. And when you do that and allow it to be above MIC for 60% of the time, you actually see effect comparable to that of the other carbapenems.
Yes, thank you. I don't think this requires a response, but I agree completely with you that there might be a compound out there somewhere that is actually a TB drug, and we just have to prove it.
Thank you very much, Andreas. Our next